Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The early decades of the 20th century presented a profound intellectual challenge to the nascent field of evolutionary biology. The elegant framework of Darwinian natural selection, which explained the gradual accumulation of advantageous traits through differential survival, required further development. A parallel revolution was unfolding in genetics, spearheaded by Gregor Mendel's meticulous experiments with pea plants, revealing the particulate nature of inheritance. Bridging the gap between these two powerful ideas, the observable, continuous variation in populations and the discrete units of heredity proved a formidable task. It was within this fertile scientific milieu that Sewall Wright emerged, a figure whose groundbreaking contributions would fundamentally reshape our understanding of evolution. Born in Medford, Massachusetts, in 1889, Wright's early life was marked by intellectual curiosity and a deep engagement with the natural world. His father, a congregational minister, fostered a love of learning and a rigorous approach to inquiry. The family's frequent moves, driven by his father's pastoral duties, exposed young Sewall to diverse environments, perhaps subtly instilling an appreciation for variability. His formal education began at Lombard College in Galesburg, Illinois, where he pursued a broad curriculum. His subsequent studies at Tufts College ignited his passion for biology and mathematics. Under the mentorship of Professor Alpheus Hyatt-Verrill, a distinguished naturalist, Wright's innate aptitude for quantitative analysis found a powerful outlet in biological problems. A pivotal period in Wright's intellectual development occurred during his graduate studies at Harvard University. While officially working in zoology under the esteemed evolutionary biologist William Morton Wheeler, Wright also delved deeply into mathematics and statistics. His time at Harvard exposed him to the burgeoning quantitative methods being applied to biological data, a nascent approach that would become his signature. It was here that he began to grapple with the complex interplay of genetic factors and observable traits, the very essence of quantitative genetics. The intellectual currents at Harvard, a hub of scientific innovation, provided the ideal environment for his nascent ideas to germinate. Upon completing his doctorate in 1915, Wright accepted a position at the Bureau of Animal Industry within the United States Department of Agriculture. This appointment placed him at the heart of practical, real-world challenges in animal husbandry. The Bureau's mission was to improve the efficiency and productivity of American livestock through scientific research and application. For Wright, this was an extraordinary opportunity. He was tasked with applying genetic principles to the improvement of economically important traits in farm animals, such as cattle, sheep, and guinea pigs. This work demanded a sophisticated understanding of how to manipulate breeding programs to achieve desired outcomes, a task that inherently required robust quantitative methods. The problems faced by animal breeders were tangible and pressing. They sought to increase milk production in dairy cows, enhance wool quality in sheep, and improve the overall robustness and fertility of their herds. These traits, however, were influenced by numerous genes, each with a small effect, interacting with each other and with environmental factors. This complexity meant that simple selective breeding based on observable phenotypes was often inefficient and unpredictable. Breeders needed a way to understand the underlying genetic architecture of these quantitative traits and to predict the outcomes of different mating strategies. At the Bureau, Wright was an active participant in experimental design and data analysis. He worked with large data sets generated from breeding experiments, meticulously analyzing the performance of different breeds and crosses. His early work focused on understanding the effects of inbreeding and crossbreeding on the variability and uniformity of animal populations. Inbreeding, the mating of related individuals, tends to increase homozygosity, potentially leading to the expression of deleterious recessive alleles. Crossbreeding can introduce heterozygosity and sometimes results in hybrid vigor or heterocies, where the offspring exhibit enhanced traits compared to their parents. Quantifying these effects and understanding their genetic basis was crucial for developing effective breeding strategies. Wright's early research at the Bureau of Animal Industry provided the empirical bedrock for his theoretical innovations. The need to make sense of the complex inheritance patterns of traits like size, weight, and disease resistance in livestock provided a direct impetus for his development of sophisticated mathematical models. He recognized that to truly advance animal breeding, a deeper theoretical understanding of population genetics was required, one that could integrate Mendelian principles with the statistical realities of continuous variation. His work here was foundational, laying the groundwork for the theoretical framework that would soon define his career and revolutionize evolutionary biology. The practical demands of improving livestock were, in essence, the crucible in which his revolutionary ideas were forged. Sewall Wright's early immersion in the practical challenges of animal husbandry at the Bureau of Animal Industry provided a fertile ground for his burgeoning mathematical genius. The prevailing aim in livestock breeding was the enhancement of desirable traits, increased milk yield in dairy cows, greater muscle mass in beef cattle, or improved wool quality in sheep. Breeders observed that offspring often exhibited a blend of parental characteristics, sometimes exceeding the average, and at other times falling short. The fundamental question that occupied the minds of these practical scientists, and which Wright would soon address with unparalleled rigor, concerned the underlying mechanisms governing this inheritance of continuously varying traits. How could one reconcile the discrete, particulate nature of Mendelian genes with a smooth, quantitative spectrum of observable phenotypes. This was a significant intellectual hurdle for evolutionary theory, particularly for Darwin's concept of natural selection acting upon gradual variations. Darwin himself had grappled with the problem of inheritance, proposing a theory of pangenesis involving gemmules that carry hereditary information from all parts of the body. This idea proved difficult to reconcile with the emerging evidence from Gregor Mendel's work on pea plants, which suggested inheritance operated through discrete units, what we now call genes. The challenge for biologists and mathematicians alike was to bridge this apparent chasm between Mendelian discrete inheritance and the observed reality of continuous variation in populations, a variation that seemed indispensable for the gradual evolutionary changes Darwin envisioned. Wright's initial foray into this complex domain involved developing mathematical models to describe the effects of inbreeding and crossbreeding on populations. He recognized that the statistical consequences of mating patterns were profound and could be quantified. At its core, his approach was to track the probabilities of individuals possessing identical alleles inherited from a common ancestor. This led him to develop methods for calculating coefficients of inbreeding, measures that quantified the likelihood of an individual inheriting two copies of the same allele through descent from a shared ancestor. Consider the problem of inbreeding. When individuals within a population mate with relatives, there's an increased probability that they will share alleles inherited from a common grandparent or great-grandparent. Wright's genius lay in devising a recursive method to calculate this probability across generations. He expressed this relationship using a system of linear recurrence equations. For instance, if we consider the coefficient of inbreeding of an offspring, denoted by f subscript t, in generation t, it could be related to the inbreeding of its parents in generation t-1. If the parents were themselves inbred with coefficients f subscript p1 and f subscript p2, and if they were unrelated to each other, meaning their inbreeding coefficients did not contribute to each other's inbreeding, then the offspring's inbreeding coefficient could be expressed as a function of the average inbreeding of the parents and the probability of receiving identical alleles from each parent. Wright's formulation elegantly captured the gradual increase in homozygosity that accompanies inbreeding. He introduced the concept of the path coefficient, a powerful tool that allowed him to visualize and quantify the direct and indirect causal influences between different variables in a system. For inbreeding, a path coefficient could represent the correlation between the alleles an individual receives from its mother and the alleles it receives from its father, a correlation that increases with the degree of relatedness of the parents. His seminal 1921 paper, Systems of Mating, published in the journal Genetics, laid out these foundational concepts with remarkable clarity and mathematical precision. Here, Wright explored various mating systems, including random mating, self-fertilization, brother-sister mating, and parent-offspring mating. For each system, he derived equations describing the rate at which homozygosity increased and heterozygosity decreased. For example, under self-fertilization, where an individual mates with itself, the coefficient of inbreeding increases by 1/2 in each generation. This means that after a few generations of selfing, a population would become almost entirely homozygous. Brother-sister mating, a more intense form of inbreeding, leads to a much faster increase in the inbreeding coefficient, reaching high levels within a few generations. These models provided a quantitative framework for understanding the genetic consequences of mating choices in real populations. For animal breeders, this meant understanding how different mating strategies could accelerate or decelerate the fixation of desirable or undesirable traits. Wright's work demonstrated that inbreeding, while potentially useful for rapidly fixing homozygous desirable genes, also carried the risk of increasing the frequency of deleterious recessive alleles, leading to inbreeding depression. His approach to crossbreeding was equally insightful. Crossbreeding, the mating of individuals from different populations or breeds, was often employed to combine advantageous traits from different lineages. Wright's statistical methods could quantify the expected genetic makeup of the offspring and the subsequent generations. He analyzed how the blending of different genetic backgrounds affected the variance of traits within the population. His models highlighted that the initial cross of two distinct inbred lines often resulted in a population with high heterozygosity and reduced variance for certain traits, a phenomenon known as hybrid vigor or heterosis. Subsequent generations, if interbred randomly, would experience a breakdown of this heterozygosity, and the variance would increase as different combinations of alleles segregated. Wright's early work was revolutionary due to its integration of Mendelian genetics with statistical analysis and a clear understanding of population dynamics. He built a quantitative theory that could predict the genetic structure of populations under different conditions. His use of path coefficients provided a visual and analytical language for understanding complex causal relationships, a language that would prove invaluable in his later, more ambitious theoretical endeavors. These early models, born from the practical necessities of improving livestock, established Wright as a pioneer in the nascent field of population genetics, offering a rigorous mathematical foundation for understanding the very fabric of heredity and its impact on the evolutionary trajectory of populations. They provided the essential tools to dissect the genetic architecture of populations, moving beyond simple observation to precise quantification. The fundamental challenge when seeking to understand the mechanisms of evolution lay in reconciling the discrete, particulate nature of inheritance, as revealed by Mendel, with the continuous variation observed in populations. Early evolutionary thought often grappled with how small, incremental changes could arise and become fixed, especially when considering the potential for chance to influence the fate of new traits. In the early 20th century, the prevailing view, heavily influenced by Darwin and later refined by biologists, often emphasized gradual, deterministic processes. A crucial question remained: what role did random chance play, particularly in the subtle fluctuations of genetic makeup within populations? This was the intellectual terrain Sewall Wright began to meticulously chart, moving beyond mere observation to construct a predictive, quantitative framework for evolutionary change. Wright's exploration of genetic drift emerged from a deep engagement with the practicalities of animal breeding, but his theoretical insights soon transcended these origins. He recognized that even in the absence of selection, the very act of reproduction, especially when dealing with finite numbers of individuals, could lead to unpredictable shifts in the frequencies of inherited traits or alleles. Imagine a small population of, say, 1,000 individuals. If a particular allele, perhaps one conferring a subtle advantage or disadvantage, is present in only a handful of carriers, the chance of those carriers successfully reproducing and passing on that allele to the next generation is inherently probabilistic. That transmission is assured. Some individuals may fail to reproduce. Others may have fewer offspring than average. These random deviations from expected proportions, when accumulated over generations, can lead to significant changes in the genetic composition of the population, irrespective of any selective pressure. This phenomenon, which Wright would famously quantify and named genetic drift, represented a profound departure from purely deterministic evolutionary models. To grasp the magnitude of this concept, consider a simplified scenario. Suppose an allele exists alongside allele in a population, and their frequencies are initially balanced. In a very large population, the random sampling of gametes during reproduction would, on average, maintain these frequencies quite closely. However, in a small population, the sampling error becomes much more pronounced. If, by chance, more individuals carrying allele A happen to reproduce successfully or have more offspring than those carrying allele A in a single generation, the frequency of A will increase and the frequency of A will decrease. Conversely, if the carriers are luckier, a frequency will decline. Over time, this random walk of allele frequencies could lead to the complete loss of one allele and the fixation of the other, a process that occurs purely due to the vagaries of chance. This was a powerful insight. Evolution was also a narrative shaped by the inherent randomness of finite populations. Wright's genius lay in his ability to translate this intuitive understanding into precise mathematical language. He developed sophisticated methods to measure the degree of inbreeding within populations and to quantify the genetic differentiation that arises between them. Central to this endeavor was his formulation of the F-statistics, a suite of measures that provided an unparalleled tool for dissecting the genetic structure of populations. These statistics offered a way to understand the variation occurring within local breeding groups and the variation occurring between these groups. The first of these, F, measures the degree of inbreeding within an individual relative to the subpopulation to which it belongs. It quantifies the excess of homozygotes within a subpopulation compared to what would be expected under random mating. A high F value suggests that individuals are more likely to be mating with relatives than expected by chance within their local group. The second statistic, F-cent, measures the degree of genetic differentiation between subpopulations. It quantifies the proportion of the total genetic variance that is attributable to differences between these subpopulations. A high FST indicates that the subpopulations are genetically distinct, with allele frequencies differing significantly from one another. The third, FIT, measures the total inbreeding coefficient of an individual relative to the entire population. It captures the combined effects of inbreeding within subpopulations and differentiation between them. These F statistics provided a quantitative framework for understanding Wright's broader theoretical concerns about population structure. They allowed researchers to empirically assess the impact of factors like population, subdivision, migration, and random genetic drift on the genetic makeup of populations. For instance, if a species was found to have high FST values across its geographic range, it would suggest that the various populations were genetically isolated from each other, with drift playing a significant role in their divergence. Conversely, low FST values might indicate substantial gene flow between populations, homogenizing their genetic composition. Crucially, Wright's work also introduced the concept of effective population size, often denoted as NE. This was a vital refinement of the simple census population size, denoted as N. Census size counts all individuals, but effective population size reflects the number of individuals contributing genes to the next generation. Factors such as unequal numbers of males and females, variation in reproductive success among individuals, and fluctuating population sizes can all reduce the effective population size below the census size. A smaller effective population size means that genetic drift will have a stronger effect, leading to faster changes in allele frequencies and a greater loss of genetic variation. Understanding in sub-E became paramount for predicting the rate at which populations would evolve and the extent to which they would lose genetic diversity. The implications of genetic drift and the concept of effective population size were profound. They offered a mechanism for evolutionary change that operated independently of natural selection, particularly in small populations. This meant that neutral or even slightly deleterious alleles could, by chance, increase in frequency and become fixed, while beneficial alleles could be lost. This challenged the prevailing view that evolution was a purely adaptive process, highlighting the pervasive influence of chance. Wright's F statistics Born from this theoretical exploration, provided the empirical tools to investigate these processes, offering a robust methodology for analyzing population structure and genetic diversity. Their enduring relevance is evident in their widespread application today in fields such as conservation biology, where understanding population subdivision and genetic diversity is critical for species survival, and in human genetics, where they are used to infer population history and migration patterns. The ability to quantify genetic differentiation and inbreeding through these statistics offered a powerful new lens through which to view the intricate tapestry of life and its evolutionary transformations. The intricate dance of evolution, Wright perceived, involved more than the random fluctuations of genetic drift, while the fixation of slightly deleterious alleles and the loss of beneficial ones underscored the role of chance in isolated populations. A richer understanding demanded an accounting of forces that connected these populations and introduced new genetic variation. The problem of how diverse evolutionary forces interacted within structured populations presented a formidable intellectual hurdle. Wright's profound contribution lay in his development of models that integrated gene flow, mutation, and selection with his foundational framework of genetic drift. This integrated approach allowed for a more nuanced comprehension of evolutionary trajectories, particularly in populations that were not monolithic entities, but rather collections of interconnected subgroups. Central to this endeavor was Wright's conceptualization of subdivided populations. He envisioned populations as being divided into smaller, semi-isolated local populations, or demes. Within each deme, genetic drift would operate, leading to random changes in allele frequencies. However, these demes, while partially isolated, were connected. Gene flow, the migration of individuals and their genes between demes, served as a powerful counteracting force to drift. The rate at which this gene flow occurred, coupled with the intensity of selection acting on different alleles within each deme, created a dynamic equilibrium. Wright's mathematical formulation of this dynamic is elegantly captured in his work on the shifting balance theory. He proposed that evolution could proceed most effectively through a process where selection acted on variations within local populations. Drift caused these variations to become fixed or lost randomly, and gene flow then spread advantageous combinations of alleles to other populations. This interplay allowed for the exploration of vast adaptive landscapes, conceptualized as multidimensional surfaces where the fitness of different genotypes was represented by altitude. Selection, in this metaphor, was the process of climbing the nearest peak. In a large, pimictic, or randomly mating population, selection might lead to fixation on a suboptimal peak, a local optimum, rather than the globally superior one. The introduction of population subdivision offered a mechanism to escape these evolutionary cul-de-sacs. In Wright's model, drift within smaller demes could lead to novel combinations of alleles, some of which might be advantageous under local conditions or represent a stepping stone to a higher adaptive peak. If such a beneficial combination arose and increased in frequency within a deme, gene flow could then disseminate this superior genetic material to other demes. This process of interdemic selection allowed for the exploration of the adaptive landscape on a grander scale, facilitating the evolution of complex adaptations and, ultimately, speciation. The rate of gene flow became a critical parameter in this theory. When gene flow occurred at a high frequency, it tended to homogenize allele frequencies across all demes, diminishing the influence of local drift in selection. In such circumstances, the population would function as a singular large unit, impeding progress toward higher adaptive peaks. Conversely, when gene flow occurred at a low frequency, populations could become excessively isolated, leading to substantial divergence and potential inbreeding depression, while forfeiting the advantages of disseminating beneficial innovations. Wright's models investigated the intermediate range of gene flow where balance could be achieved, facilitating both local adaptation and the dissemination of successful variants. Mutation also contributed, supplying the foundational material for selection and drift to operate upon. New alleles emerging through mutation could introduce novel variations into the gene pool, presenting new avenues for adaptation. The mutation rate, while typically low, served as a consistent source of genetic novelty, ensuring the evolutionary process remained dynamic. Wright's framework recognized mutation as a continuous input into the system, influencing the allele frequencies that drift and selection acted upon. The interaction among these forces, drift, mutation, selection, and gene flow, within a structured population offered a sophisticated explanation for the evolutionary patterns observed in nature. It provided a resolution to the perceived conflict between gradual adaptation and the potential for rapid evolutionary change. The shifting balance theory proposed that evolution was a complex process, propelled by both chance and deterministic forces, operating at various scales within populations. The contemporary significance of Wright's perspectives on subdivided populations and the equilibrium of evolutionary forces remains substantial. The concept of metapopulations, which are collections of interconnected populations experiencing local extinction and recolonization, directly reflects Wright's theoretical framework. Comprehending the dynamics of gene flow between these metapopulations is vital for conservation biology. For example, preserving corridors for migration between fragmented habitats can prevent local populations from becoming overly genetically isolated, thereby safeguarding adaptive potential and reducing the risks of inbreeding. Furthermore, Wright's models provide a foundational understanding for contemporary research into population genetics and evolutionary biology. The principles governing the balance between local adaptation and gene flow are still actively investigated in diverse organisms, from microbial communities to large mammals. The study of how genetic variation is maintained within species, how new species arise, and how populations respond to environmental change all owe a significant debt to Wright's pioneering work on the complex interactions of evolutionary forces in structured populations. His theoretical constructs provided the conceptual scaffolding upon which much of modern evolutionary genetics has been built, offering a powerful lens through which to examine the intricate mechanisms that shape the diversity of life. The intricate web of cause and effect, particularly within biological and social systems, presents a persistent challenge for scientific inquiry. Researchers grapple with untangling the myriad influences that shape observable phenomena, seeking to quantify the direct and indirect pathways through which one variable impacts another. Early statistical methods often struggled to represent and analyze these complex, multivariate relationships with the precision required for robust causal inference. The prevailing approaches, while valuable, frequently offered limited capacity to dissect the nuanced interplay of multiple contributing factors, leaving a crucial gap in understanding the underlying mechanisms driving observed outcomes. Sewall Wright's profound contribution to addressing this fundamental problem arrived with the development of path analysis. This innovative framework offered a powerful visual and statistical language for dissecting causal relationships. Wright envisioned a system where complex interactions could be systematically represented, allowing for the quantification of direct effects, those occurring without intermediary steps, and indirect effects, those transmitted through a chain of intervening variables. His method employed diagrams, now universally recognized as path diagrams, where variables are represented as points and causal influences as directed arrows. Each arrow carried a coefficient, a path coefficient, which quantified the strength and direction of the causal relationship it represented. The brilliance of path analysis lay in its ability to move beyond mere correlation, which merely indicates association, towards an estimation of causal influence. Wright meticulously formulated the statistical underpinnings, demonstrating how observed correlations between variables could be decomposed into components attributable to direct effects, indirect effects, and residual associations arising from unmeasured factors or reciprocal causation. He established the rules for calculating these path coefficients, essentially providing a method to estimate the magnitude of influence along each specified causal pathway. Path analysis enabled researchers to diagrammatically represent influences such as rainfall, soil fertility, and fertilizer application when seeking to understand factors influencing crop yield. It allowed for the statistical estimation of the direct impact of each on yield, alongside any indirect impacts mediated through other variables, such as soil fertility's influence on fertilizer effectiveness. This approach offered a systematic way to test hypothesized causal structures. Researchers could propose a model, a specific arrangement of variables and causal arrows, and then use the observed data to estimate the path coefficients. The strength of the fit between the model and the data, along with the significance of the estimated coefficients, provided evidence supporting or refuting the validity of the proposed causal relationships. Wright's work provided a blueprint for building and testing causal models, a significant advance in the analytical toolkit available to scientists. The conceptual foundation of path analysis rested on the idea that any correlation between two variables could be understood as a sum of contributions from all possible causal paths connecting them, including those operating through intermediate variables. Wright's algebraic derivations showed how these path coefficients could be directly calculated from the observed correlation matrix of the variables. This allowed for a quantitative assessment of the relative importance of different causal pathways, offering insights that were previously unattainable. Consider, for instance, a study investigating the factors affecting student academic performance. A path diagram might include variables such as socioeconomic status, parental education, school resources, student effort, and ultimately, academic grades. Path analysis would permit the estimation of the direct effect of student effort on grades, while also quantifying the indirect effect of socioeconomic status on grades, perhaps mediated through access to better school resources or higher parental educational aspirations. This level of detail allowed for a much deeper understanding of how various societal and individual factors converged to influence educational outcomes. The modern relevance of path analysis is profound and far-reaching. It serves as the foundational methodology for structural equation modeling, or SEM, a powerful statistical technique widely employed across a vast spectrum of scientific disciplines, including psychology, sociology, economics, education, medicine, and biology. SEM extends path analysis by incorporating latent variables, constructs that are not directly measured but are inferred from a set of observed variables, and allowing for the modeling of measurement error. This expansion has made SEM an indispensable tool for researchers seeking to analyze complex theoretical models and explore intricate relationships between multiple variables. In fields like psychology, SEM is used to model personality structures, test theories of cognitive development, and understand the underlying mechanisms of mental health disorders. Sociologists utilize it to examine social stratification, the dynamics of social networks, and the impact of public policy. In economics, it helps in understanding consumer behavior, financial markets, and macroeconomic relationships. Medical researchers employ SEM to investigate the complex interplay of genetic, lifestyle, and environmental factors in disease etiology. In evolutionary biology, the principles derived from path analysis continue to inform the study of complex trait evolution and gene-environment interactions. The enduring legacy of Wright's path analysis lies in its ability to provide a rigorous, quantitative framework for understanding causality in systems characterized by multiple, interacting influences. It offered a systematic approach to hypothesis testing concerning causal structures, enabling researchers to move towards inferential statements about the mechanisms driving observed phenomena. The visual clarity of path diagrams, coupled with the statistical rigor of coefficient estimation, provided a powerful synthesis of graphical representation and quantitative analysis, a combination that continues to empower scientific discovery and deepen our comprehension of the world's complex systems. This methodology remains a cornerstone of modern statistical modeling, a testament to Wright's singular vision in developing a tool to illuminate the intricate pathways of influence that shape our reality. Sewall Wright's intellectual journey, marked by its relentless pursuit of understanding the intricate mechanisms of evolution, extended into the later decades of his life with a profound synthesis of his accumulated insights. Having illuminated the power of path analysis to untangle causal relationships in complex biological systems, Wright turned his considerable energies towards integrating his diverse theoretical contributions into a more unified framework for evolutionary biology. His work on human genetics, a field ripe with challenges and opportunities for quantitative analysis, became a significant arena for the synthesis. The study of human inheritance presented a unique set of problems for evolutionary theorists. Unlike the controlled breeding experiments possible with laboratory organisms, human populations offer a complex tapestry of mating patterns, migration, and cultural influences that profoundly shaped genetic variation. The prevailing approaches often struggled to disentangle the effects of these myriad factors. Wright, with his established methodologies, saw an opportunity to apply and refine his tools to this vital area. He recognized that understanding human evolution required a nuanced appreciation of how genetic drift, mutation, selection, and gene flow interacted within populations of varying sizes and structures. His research in this domain aimed to quantify the relative contributions of these evolutionary forces to the observed genetic diversity within and between human populations. A key aspect of Wright's later work involved the meticulous application of his quantitative methods to real-world genetic data. He grappled with the complexities inherent in human populations, such as assortative mating, which deviates from random mating and can alter allele frequencies and genotype distributions in predictable ways. Wright's path analysis provided a powerful lens through which to examine these deviations, allowing him to model the flow of genetic material and the influence of social structures on evolutionary trajectories. His analyses sought to quantify the impact of factors like endogamy in isolated communities or the genetic consequences of societal stratification. These represented a rigorous attempt to build a predictive science of human heredity grounded in the principles of population genetics. This period also saw Wright engaging deeply with the broader evolutionary synthesis, a monumental intellectual movement that sought to reconcile Mendelian genetics with Darwinian natural selection. While figures like Fisher and Haldane had already made significant strides in developing mathematical models of evolutionary processes, Wright's perspective offered a unique emphasis on the role of population structure and random sampling effects. His concept of the shifting balance theory, which posits that evolution progresses through periods of small, isolated populations experiencing drift, followed by periods of interbreeding and selection, provided a crucial perspective on models that emphasized continuous, large population dynamics. He argued that genetic variation could be maintained and novel adaptations could arise more effectively through a dynamic interplay of these forces, a notion that resonated with observations of speciation and adaptation in the natural world. Wright's discussions with his contemporaries, particularly Ronald Fisher and J.B.S. Haldane, were often characterized by vigorous intellectual debate, yet they also underscored a shared commitment to building a robust mathematical foundation for evolutionary theory. Fisher, for instance, often focused on the power of selection in large populations, while Wright consistently highlighted the critical role of random genetic drift, especially in smaller populations. He saw drift as a potent evolutionary force capable of fixing alleles that might be disadvantageous under pure selection, and thereby creating new genetic starting points for adaptive evolution. This nuanced view of the interplay between deterministic selection and stochastic drift was a hallmark of Wright's contribution to the synthesis. The problem of unifying these disparate evolutionary forces, mutation, selection, migration, and drift, into a coherent and comprehensive theory was a central challenge of the era. Wright's approach was to develop mathematical models that could simultaneously incorporate these elements, allowing for the estimation of their relative importance in specific scenarios. His development of the coefficient of inbreeding, often denoted by the symbol F and its extensions, such as the F statistics, specifically F sub st, F sub is, and F sub it, provided a powerful quantitative toolkit for measuring genetic differentiation among populations and the inbreeding within them. These statistics allowed researchers to partition total genetic variance into components attributable to different levels of population structure, offering a direct measure of the impact of gene flow and genetic drift. F subset, for example, quantifies the proportion of total genetic variance that is due to differences between subpopulations. A high F sub ST indicates substantial genetic differentiation, suggesting limited gene flow and strong local selection or drift. Conversely, a low F sub ST implies that subpopulations are genetically similar, indicating significant gene flow or recent divergence. Similarly, F sub measures the deviation from random mating within subpopulations, often reflecting the effects of inbreeding or outbreeding. F sub IT captures the total inbreeding of individuals relative to the entire population. These measures provided a concrete, quantitative way to assess the impact of Wright's shifting balance theory in empirical studies. His later work also saw him grappling with the genetic underpinnings of complex traits, those influenced by multiple genes and environmental factors. The challenge here was immense: how to disentangle the additive genetic effects from dominance, epistasis, gene-gene interactions, and gene-environment interactions. Wright's path analysis, originally conceived for simpler causal systems, proved remarkably adaptable to these more intricate scenarios. He developed methods for estimating heritability, the proportion of phenotypic variance attributable to genetic factors, and for dissecting the components of genetic variance. This was crucial for understanding how selection could operate on traits that followed Mendelian patterns, a common situation in natural populations. The intellectual landscape of evolutionary biology during this period was dynamic, with ongoing discussions about the relative importance of various evolutionary forces. Wright's contributions, particularly his emphasis on the adaptive landscape and the shifting balance theory, offered a qualitative and quantitative framework that complemented the work of his peers. He envisioned evolution as a process unfolding on a multidimensional surface, where peaks represented adaptive genotypes and valleys represented less fit ones. Genetic drift, in his view, could allow populations to escape local adaptive peaks and explore new regions of the landscape, potentially leading to novel adaptive breakthroughs. This visual metaphor, underpinned by rigorous mathematics, provided a powerful conceptual tool for understanding the contingent and sometimes unpredictable nature of evolutionary change. Wright's dedication to synthesizing these complex ideas into a coherent, overarching theory of evolution was a testament to his intellectual perseverance. He sought to bridge the gap between theoretical models and empirical observation, constantly refining his mathematical tools and applying them to diverse biological problems. His later career was characterized by a deep engagement with the fundamental questions of how life evolves, driven by a desire to understand the interplay of all major evolutionary forces within the context of real-world populations. This period of synthesis solidified his position as one of the most influential architects of modern evolutionary thought. Sewall Wright's intellectual edifice stands as a cornerstone of modern evolutionary biology, its influence radiating outward to shape fields as diverse as conservation genetics, quantitative genetics, and evolutionary developmental biology. His theoretical frameworks, meticulously constructed over decades, provided a sophisticated mathematical language for understanding the intricate dance of life's variations and adaptations. The shifting balance theory, a profound conceptual leap, offered a dynamic perspective on how populations navigate the complex landscape of fitness, moving beyond static equilibrium models. This theory illuminated the crucial role of random genetic drift in facilitating escape from suboptimal adaptive peaks, thereby enabling exploration of novel evolutionary pathways. This mechanism, deeply embedded in mathematical formulations of population structure and gene frequency change, provided a powerful lens through which to view the contingent and often surprising trajectory of evolutionary innovation. The elegance of Wright's approach lay in his ability to imbue abstract mathematical models with biological reality. He perceived populations as intricate mosaics of interacting subpopulations, each subject to different forces of evolutionary change. His work on migration and subdivision revealed how localized genetic differences, amplified by drift and selection within isolated groups, could serve as reservoirs of novel adaptations. These localized innovations, when reintroduced to the broader population through migration, possessed the potential to spread and fundamentally alter the adaptive trajectory of the species. This nuanced understanding of population structure and connectivity provided a framework for explaining patterns of biodiversity and speciation that had long eluded simpler explanations. Wright's pioneering development of path analysis offered a potent tool for dissecting complex causal relationships within biological systems. This statistical methodology allowed researchers to untangle the interwoven influences of multiple genetic and environmental factors on phenotypic traits. By representing these relationships as a system of directed paths, Wright provided a visual and quantitative means to estimate the direct and indirect effects of each variable. This approach proved invaluable in quantitative genetics, enabling the estimation of heritabilities and genetic correlations with unprecedented precision. It allowed scientists to move from observing correlations to inferring underlying causal pathways, a critical step in understanding the genetic architecture of complex traits. The enduring relevance of Wright's contributions is perhaps best illustrated by their continued application in contemporary biological research. Conservation genetics, tasked with preserving biodiversity in the face of rapid environmental change, relies heavily on Wright's insights into population structure, genetic drift, and inbreeding. Understanding how small, Isolated populations are vulnerable to loss of genetic diversity and detrimental effects of inbreeding. Concepts rigorously explored by Wright is essential for designing effective conservation strategies. His work informs decisions about population management, habitat connectivity, and the genetic rescue of endangered species, underscoring the practical import of his theoretical explorations. Furthermore, the field of evolutionary developmental biology, or Evo, finds a conceptual resonance with Wright's emphasis on the interplay between genetic variation and developmental processes. Wright's recognition that evolutionary change is contingent upon the available genetic variation and the developmental pathways that translate genotype into phenotype aligns with the core tenets of Evo. The exploration of how changes in developmental genes can lead to novel morphologies, a central theme in Evo, represents a modern manifestation of the search for the sources of evolutionary innovation that so captivated Wright. His foundational work on the genetic basis of quantitative traits also provides a crucial backdrop for understanding the genetic underpinnings of developmental plasticity and innovation. Wright's legacy is deeply embedded in the statistical methods employed across the life sciences. Path analysis, his groundbreaking contribution, has evolved into structural equation modeling, a ubiquitous tool for analyzing complex systems in fields ranging from psychology and sociology to ecology and epidemiology. The ability to model and test intricate causal hypotheses, a capability unlocked by Wright's early work, remains a vital component of scientific inquiry. His insistence on rigorous mathematical formulation and empirical validation set a standard for the integration of theory and evidence that continues to guide scientific progress. The sheer breadth of Wright's intellectual output, spanning population genetics, evolutionary theory, biometrics, and statistical methodology, solidifies his position as one of the titans of 20th century biology. He provided the mathematical scaffolding upon which much of modern evolutionary understanding is built. His ability to conceptualize complex biological processes through the elegant precision of mathematics allowed for a deeper, more quantitative apprehension of evolution's mechanisms. The exploration of genetic drift, gene flow, mutation, and selection, and their intricate interactions within structured populations was given unparalleled clarity and predictive power through his analytical frameworks. Wright's unwavering commitment to understanding the grand sweep of evolutionary history, from the molecular level of gene frequencies to the macroscopic patterns of speciation and adaptation, represents a singular achievement. He demonstrated that profound biological questions could be illuminated through the rigorous application of mathematical reasoning. His theories, initially abstract and theoretical, have proven remarkably robust and adaptable, continuing to provide essential insights into the processes that shape the diversity of life. The intellectual journey of Sewall Wright, marked by deep contemplation and mathematical ingenuity, offers a compelling testament to the power of theoretical science to unravel the most fundamental questions of existence. His work remains an indispensable guide for anyone seeking to comprehend the intricate, dynamic, and often surprising journey of evolution.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.